All right, hello everyone. Today we are diving deep into what is essentially the engine room of the microbial world, bacterial metabolism. Now, this isn't just going to be a boring list of chemical reactions. Think of it more as a sophisticated biochemical toolkit. It's what allows bacteria to absolutely conquer every conceivable niche on our planet, from the crushing depths of the ocean to the inside of our own guts. It's their ultimate survival guide, and it's all written in the language of chemistry. So let's map out how we're going to tackle this. Okay, here's our game plan. First up, we'll cover the two fundamental sides of metabolism, that constant balancing act of breaking things down and building things up. Then, we're going to ask a really basic question. What do bacteria actually eat? That'll lead us right into how we classify their incredibly diverse appetites. After that, we'll get into the real nitty-gritty, the mechanics of how they generate energy. And of course, we'll look at how they then use that energy to build the very fabric of life. To wrap it all up, we'll connect everything back to the real world and talk about why all of this really matters.
Right, first things first. At its absolute core, all metabolism, and I don't care if you're a bacterium or a blue whale, is a balancing act. It really boils down to two opposing but perfectly interconnected processes. You've got one process that's breaking molecules down to release energy, and another that's using that exact same energy to build new molecules for growth and repair. And this slide lays out that balance perfectly. So on the left, you have catabolism. I like to think of this as the cell's demolition crew. It smashes up complex compounds like glucose in what we call an exergonic process, meaning it releases energy which gets captured as ATP. But here's the crucial bit. It also generates this pool of 12 essential precursor metabolites. These are just small carbon skeletons, but they're the fundamental building blocks for absolutely everything else. Then, on the right, you've got anabolism. This is the construction crew. It's all about biosynthesis. This is an endergonic process, so it requires energy. It uses that ATP to assemble those precursor molecules into complex structures like proteins and cell walls. So the main thing to get here is this. Catabolism doesn't just provide the energy for anabolism, it provides the raw materials too. It's a beautifully efficient system. Okay, so we've got the basic principle down. Now let's get into the specifics. Bacteria have this absolutely incredible diversity in what they actually consume to power these metabolic engines. I mean, it's wild. To make any sense of it, we classify them based on where they get two essential things. Carbon, the backbone of life, and energy. So first up, we've got the autotrophs. You can think of these as the ultimate self-starters of the microbial world. They don't need to go around eating complex organic molecules that other organisms have already made. Nope. They just take the most basic, inorganic form of carbon they can find, we're talking carbon dioxide straight from the atmosphere, and use that as the raw material to build all the complex organic molecules they need to live. Pretty impressive stuff. And then you've got the other side of the coin, the heterotrophs. Now, these organisms, which, by the way, includes us and a ton of the bacteria we study in the lab, are the consumers. They can't do that cool trick of building from CO2. Instead, they have to get their carbon by eating preformed organic molecules. Things like sugars, fats, and proteins that have already been put together by other organisms.
But wait a second. Getting carbon is really only half the story, right? I mean, having a pile of bricks is great, but you need energy to actually build the house. To assemble those carbon blocks into cellular structures, you need a constant supply of energy. So the big question is, where does that energy actually come from? And that brings us to our second major way of classifying these microbes. Okay, this diagram here maps out the main energy sourcing strategies, and it's super useful. Let's walk through it. At the very top, you see the first big split. If an organism gets its energy from light, it's a phototroph. Simple enough. But if it gets its energy from chemical compounds, it's a chemotroph. Now let's follow that chemotroph branch down because it gets even more interesting. It splits again based on the type of chemical. If the bacterium is oxidizing, which just means it's stripping electrons from organic chemicals like glucose, it's a chemoorganotroph. Our good old lab workhorse E. coli is the classic example here. But, and this is where it gets really cool, if a bacterium can oxidize inorganic chemicals, it's a chemolithotroph. Look at the example, Thiobacillus thiooxidans. This thing can get all the energy it needs from hydrogen sulfide or even just elemental sulfur. It literally eats rocks. It's amazing. But no matter which path they take, light, organic chemicals, or inorganic chemicals, the end goal is always the same. Generate ATP, the cell's universal energy currency.
All right, there's one last really critical way we classify bacteria, and that's their relationship with oxygen. It's a spectrum, really. At one end, you have an obligate aerobe. This guy absolutely has to have oxygen to survive. A microaerophile also needs oxygen, but only at low levels. Too much is toxic. Then, at the complete opposite end, you have the obligate anaerobe. For these guys, oxygen isn't just unnecessary, it's actually poisonous. And that's usually because they lack key protective enzymes like catalase, so oxygen radicals just tear them apart. But the most metabolically flexible of the bunch is the facultative anaerobe. This one's clever. It prefers to use oxygen when it's around, because that gives it the biggest energy payoff. But critically, it has a metabolic plan B. It can switch to anaerobic strategies to survive when there's no oxygen. That flexibility is a huge survival advantage. So that plan B I just mentioned for facultative anaerobes, well, that leads us perfectly into our next section. We're going to look at the two major strategies bacteria use to squeeze ATP out of their food: respiration and fermentation. This table does a great job of breaking down the comparison. Look at that first row, ATP yield. The difference is just staggering. Respiration is your high-efficiency engine. It completely oxidizes a molecule of glucose and cranks out up to 38 ATP. Fermentation, on the other hand, is like a tiny, low-power backup generator. It only gives you a measly 2 ATP. So you have to ask, why would any cell bother with it? Well, the answer is in that second row, the final electron acceptor. In respiration, the electron transport chain is like a conveyor belt, and at the end, it needs to dump its electrons onto something. That's the external or exogenous acceptor, which is usually oxygen. If there's no oxygen, the whole belt grinds to a halt. Fermentation is a clever workaround. It uses an internal or endogenous acceptor, something like pyruvate, just to offload those electrons so that glycolysis can keep taking over. Yeah, it's wildly inefficient, but it's an absolute lifeline that lets the cell keep making some ATP when oxygen just isn't an option.
Okay, so the cell's done the hard work, it's run its catabolic pathways, it's got a nice stash of ATP, and it's got that all-important pool of precursor molecules. So now, it's time for the construction phase. Let's take a look at how bacteria actually use these resources in anabolic pathways to build the very fabric of the cell.
What this really shows is the incredible efficiency of bacterial metabolism. You see, a cell doesn't run two completely separate systems for breaking stuff down and building stuff up. That would be a waste. Instead, the central catabolic pathways, the ones breaking down glucose, are tapped directly for anabolic precursors. So, for example, sugars needed for the peptidoglycan cell wall are pulled right out of glycolysis. And this is really important. The carbon skeletons for all 20 amino acids are siphoned off from various intermediates within glycolysis and the TCA cycle. The exact same principle applies for building nucleotides and fatty acids. It's not just an assembly line, it's a complete masterclass in biochemical economy. Absolutely nothing is wasted. So we've taken a pretty deep dive into the biochemical nuts and bolts here, but let's pull back for a second and look at the big picture. Why is it so important for us as microbiologists to really get this stuff on a granular level? Well, because this isn't just some academic exercise. It has profound real-world implications. And this quote really brings us full circle. The incredible metabolic diversity of bacteria is not just a key to their survival, but a powerful tool for human innovation. You see, from the bacterium's point of view, the goal of all these pathways is just to create the building blocks for growth. Simple. But for us, understanding this process is about so much more than just watching bacteria grow on a plate. It's about seeing their entire metabolic playbook as a library of powerful tools that we can learn to use. And this really is the key thought I want to leave you with. Once we understand these pathways, we can start to manipulate them. Think about it. We can exploit their unique metabolic weaknesses to design brand new antibiotics. In industrial biotech, we harness their fermentation pathways to produce everything from biofuels to life-saving drugs. In bioremediation, we can literally deploy chemolithotrophs, those rock eaters, to clean up toxic industrial waste. Even in the clinical lab, we use their metabolic fingerprints to figure out exactly what pathogen is causing an infection. So the question we should be asking isn't just how do bacteria survive, it's how can their survival strategies solve our problems? Their incredible metabolic diversity is really our greatest opportunity.